Patrick doing is it's everybody that we're working with. And so I want to just show you what we had this summer, for an example. So Kelsey is my daughter. She actually ran the lab. I think out of the 9 weeks, she got out of the lab one day, right? She got in the boat one day, and that was a special day for her to get out of the lab and get in the boat. But there was so much going on, as Patrick will explain. Her husband, Chris, was another guy that just did most of the field work. I'll talk about Sydney and Patrick in a minute. Matt Schuling just graduated, double major from Hope College. Dan Clyde is a senior this year. He's here, Hope College, a biology major. Annette Dobrzinski is still working in her lab because we have some research that's still going on once a week, and she's a retired teacher and lives up in Honor. And then Brooke is a PhD student with Patrick at the University of Alberta. Sydney is a real, also key piece. She's been there 5 years now, and she's near her finishing, but she's been very instrumental. You'll see her name on the publications. She's also a PhD person with Patrick. And then I can't say enough, and I probably don't say enough, about our collaboration with Patrick Hannington. And I can't imagine what we wouldn't have done these last 3 years with Freshwater Solutions if it hadn't been for our collaboration with Patrick. And I just thought of this too, that I don't think a lot of people realize. So I asked him to document how much money he donated just this year, just for swimmer's itch work. And it was $60,000 that came from Canada to Northwest Michigan for projects that we're working on. And that's not even the enteric bacteria stuff. And so we owe a lot to his generosity, certainly his knowledge base and the people he knows and the resources he has. I just can't say enough and speak highly enough of Patrick. He's here, came in Thursday and leaves Tuesday, so he always comes in during the summer. At least he has every summer or fall. And so he's here. It's always a big time. And so what we try to do is just gather people who have all been positive and supportive of what we're doing, and gather them when Patrick's around, talk a little bit about what's going on, and maybe what the future looks like. And so that's what we're doing here. So I'm not going to give a lot. We don't have a lot of final things with swimmer's itch reports. I know that's what a lot of you are waiting. What do we find? What do we find? We have some things, but we're getting there. It'll go quite quickly now that we've had a chance to spend a week together. So anyway, Dr. Patrick Hannington.
Thanks, Ron. I was telling Ron that this is the first time I think I've been coming to Michigan since 2016, sometimes twice a year, and this is the first time it's rained since I've ever been here. So now I know it rains here, but yeah, I've been very sort of happily surprised with the weather up until this point. So thanks, Ron. Like Ron said, all of these people really are the ones who do a bunch of the work, and so as Ron said, we at the University of Alberta in my lab we do a bunch of qPCR work, but all of that, I think what my hope at the end of this presentation is to show you that none of the qPCRs that we do happen if Freshwater Solutions and the community volunteers that are collecting samples actually collect those samples. And that's actually the most critical part of doing these qPCR programs, looking for things like swimmer's itch or enteric bacteria, is you have to collect samples, and the samples have to be done in a way that allow us to learn something about the targets that we're trying to find. And so we've learned over the years, and Sydney, as Ron said, her thesis is getting close to being completed now. She's a PhD student in my lab. Her thesis is based off of thousands of samples collected all over the place by people in the community. And when you put it into the perspective of how many samples she's got in her thesis, she would never have been able to collect that many samples to analyze. And so my point that I want to make sure you all get by the time this is over is that these kinds of programs, to get the kind of data that we can get from things like swimmer's itch or enteric bacteria, rely on community partnerships. And that's what I'm going to talk about today. So I'm going to try to keep this around an hour, and we'll do questions and stuff like that once I'm all finished, I think. That might be the easiest way to do this, but if you have something really burning, feel free to put up your hand, and I'll try to make sure I'm looking out on the crowd every once in a while, not at the screen. But I want to talk to you a little bit today about work that we've been doing to source trap microbial targets in recreational water using a community-based monitoring approach. And this term, community-based monitoring, this is the word that we use. Other words for this are things like citizen science or community-based or community participatory research. These are all sort of the same sort of synonyms for this approach, where you're really relying heavily on a partnership between researchers, potentially government partners or NGOs, and community members to actually address large questions in a significant way. And I'll explain a little bit about this as we go through. So for today's talk, I have a sort of a really general outline. I'd like to introduce you to community-based monitoring, and I would like to introduce you to qPCR, because I'm pretty sure that not everyone in the room knows all the intricacies of qPCR, and I don't expect you to know the intricacies when we're done, but I'll at least tell you a little bit about how it works. And I would like to show you a case study of how to develop a community-based monitoring program. And to show you that case study, I would like to use some of the sperm-rich work that we've been doing over the last 3 years. And this is really what we can learn from an army of citizen scientists working towards the same goal. And that's really how we've structured a lot of the sperm-rich research that we've been building upon each year. So I'm not going to give you much sperm-rich detail here, but I'm going to try to tell you this story really succinctly, just so that you can get a sense for what we started with and what we're now working at and how it's going to go forward. And then finally, a lot of you are here because of this interest in enteric bacteria, and this is what I would like to finish with. Is we do a lot of enteric bacteria work in Alberta, where the province in Canada that I work in. And we started translating some of that work here in Michigan, and I think this is some of the stuff that we see being sort of the way forward for water monitoring programs here. So I would like to end with that and talk a little bit about some of the really fantastic programs you already have in Michigan for water monitoring at a recreational level, and then also how we think we could build upon that awesome net system already with some community-based monitoring programs. So hopefully you can see here, you probably recognize this place, Michigan. This is Alberta overlaid on top of that. So where I come from, it's a big province in Alberta, covers a lot of space. Up on this end of the border, the northern border, we have the Yukon. Alaska is just kind of right over here. And so we have a pretty big range. Down here would be borders with Montana. And so we have a problem with water monitoring in Alberta. As Ron can attest to, he's been there. We have lakes that you would never swim in. You here have amazing lakes compared to the lakes in Alberta. And so our lakes are gungy usually. They're shallow. They're very eutrophic, which means they have lots of algae and stuff like that. But we also have lakes in the mountains that are really pristine. They're beautiful. And that ranges depending on where you are in the province. And so like Alberta, Michigan is also large. It has over 11,000 inland lakes, and you have thousands of stormwater catchment areas. And this is similar to Alberta. You have way more lakes actually than Alberta has in this littler space. So you're the land of lakes. There are a lot of different types of targets that we try to monitor for recreational waters, and this means that you have to have tests that can detect all those different targets, and that is a challenge. When you're thinking about looking for enteric bacteria or sperm-rich or invasive species, all those things have to have a test that you can apply. And so some of those targets, like enteric bacteria, will have policies associated with them. And here in Michigan, it's no different. You have policies and guidelines that are associated with a lot of the enteric bacterial targets that are allowable levels of these different organisms to have in a water sample before you may, say, have a state posting on a beach to close the beach to recreational use, or they might trigger some sort of thing where you have to do an investigation. The other thing, I think most of you probably realize this, that water is dynamic and can change quickly. And that means that the targets that you're interested in looking for, or the targets that you are monitoring, can change really quickly, right? If you have a rainfall event like we have right now, the water sample you collect today is going to be very different than the water sample you collect after 2 weeks of sunshine. So these are all things that really give us challenge when we think about monitoring water, either wastewater or stormwater or recreational water or drinking water. All these things can be impacted by all these different factors. And our solutions to this have been to start thinking about involving community members much more in our research and monitoring programs. And so our goals have been really to try to start unifying testing methodologies to allow us to use a single method to detect all the different organisms that we want to find in water. And this isn't just us that are doing this. There's a lot of people that are working towards this kind of a goal. maybe what's more unique to us is that we are also working towards trying to decentralize lab testing. And so for your beach monitoring program in Michigan, you probably have people who collect water samples, sometimes routinely, probably not very routinely, from public beaches. Those water samples are analyzed in a core facility lab. In Michigan, you've actually started to decentralize those labs. So you may have regional labs that do something like coli alert or entero alert to detect enteric bacteria in your water sample. And then that gets reported back. And we propose that decentralizing is a really important way to help more routinely monitor water and get more frequent updates on water quality. One of our big things is to train and trust citizen scientists, and this is a big theme that's going to run through this whole presentation, is the importance of people like you and making sure that samples that are collected are done consistently and that everything is done properly, but also making sure that we trust that once everyone is trained that you are doing things properly, because you can't have an element of distrust because then you don't believe the results, right? So ultimately our goal is to really democratize water monitoring and start really expanding scope, scale, frequency, all these elements that allow us to answer really big questions about water. So when we think about what we want to monitor in water, we break down organisms in water into a couple different categories. Obviously there are health-related targets, there are targets that are maybe more important for environmental reasons, and then there are some that have an economic impact. And lots of them bridge between all of this, right? We know, for example, that swimmer's itch probably touches on all of these things with having a health impact. Certainly, for those of you that have had a bad swimmer's itch case, you know that it is unpleasant. But there's maybe an economic impact there, where if you have a lake that has routine swimmer's itch reports, maybe you have Airbnbs on the lake that aren't used as frequently because people don't want to go to a lake with lots of swimmer's itch, or it's harder to sell your property because the swimmer's itch is really bad in a certain spot. Things like that. We see that in Alberta actually more frequently with cyanobacteria, which is our big problem. You've heard of that, I think, as well, in places like Lake Erie and stuff like that. But then we have other things, fecal coliforms, enteric bacteria that can be economic, but certainly health. Some environmental bacteria like Legionella, health and an environmental element. And then we have lots of invasive species and other types of plants, invasive plants and fish that tend to be more on the economic and environmental side.
How are you defining environment?
Just any environment. So it could be in-premise plumbing as an environment, it could be a lake as an environment, it could be any.
When it touches on environment, that
Yeah.
means that it has some type of impact on it.
Sure, yeah, or the environment is impacting it.
Okay, so
Yeah.
we're saying here that swimmer's itch could have a negative environmental impact?
Oh, absolutely, yeah. Yeah, you could
I
have,
wasn't aware of that.
yeah, you could have, I mean, the parasites that cause swimmer's itch can cause problems with waterfowl. You can have changes in parasite biodiversity. Parasites themselves are significant sources of food for filter feeding organisms. And so lots of different things touch on what the importance of parasites are to the ecosystem. Yeah. So I would say even more broader than swimmer's itch, you could say all parasitic trematodes have an environmental impact.
I mean, they have the plates there.
Yeah.
Right.
Certainly, yeah. Yeah, so our goal is to move away from testing for things like, say, cyanobacteria, testing for that using a visual inspection for cyanobacteria presence or absence. And then you might test for fecal coliforms using a coli alert test. And you might test for Legionella using qPCR. The advantage here is that qPCR as a technique, which is really a DNA-based detection tool, can detect all biological organisms. So as long as you know enough genetic material or genetic information about an organism, you can design qPCR tests that allow you to detect parasites in the water. This is a parasite that causes whirling disease, invasive mussels, cyanobacteria, swimmer's itch, and you can add every other one of these things on that list as well. And you can even use different types of DNA. A big tool that's being used these days is called eDNA, environmental DNA. That's being used a lot to detect invasive species or species at risk. So for the places, for example, in the Great Lakes where you have invasive fish, eDNA is being used very frequently to detect where those fish are impacting. So this gets at this idea of unifying testing methodologies, and this is what I want to talk to you about a little bit more in detail, is the importance of qPCR as a tool for monitoring. So before I touch on that, I want to tell you what community-based monitoring is and how it can work. So we see a few different models of community-based monitoring, and these are things that I think probably all of you have seen before. Model A is sort of a very top-down approach, where a researcher designs a project, interprets results, and then they can recruit community partners to collect samples, and the data often will reside with the researcher. many CBM water monitoring programs fit into this model. Ron has already told me about a few where some of you may have been involved, where you get a Secchi disc or a little kit and you collect a water sample with a bottle and you measure visibility in the water, you collect a water sample, you send it off somewhere, and then you might get a report at the end of the year, or you might be able to access a report at the end of the year to see what came of all that. But that's really, you are being a sample collector for a bigger program that you're not really having any input on in terms of designing what you're doing, right? You probably don't get to tell the guy who tells you to do a Secchi depth, I want to do this many Secchi depths and I'm going to send you this many bottles of water. They just say, do this, and you do it. Model B is where there's a researcher that designs the project and interprets results. The community partner collects the samples, and then the data is routinely shared, and so there's more connection there. And this has really been sort of how our freshwater solutions swim research program has been to date, where we've relied on freshwater solutions. My lab has relied on freshwater solutions as a sort of community partner, but then freshwater solutions has also relied on some of you to help collect samples, and then there's annual reports and more communication between the partners to talk about the results. The final one, which is, I'll give you a little heads up, this is the one that we're trying to move towards here in Michigan, is Model C, where we have a researcher and a community partner that designs a project. The researcher helps interpret the results with the partners. The community partner collects the samples typically, but the data is shared and the interpretation is done together. And this is an important distinction because here the research itself is being designed by both partners. And so we want to make sure that the community partners are having as much input on the research questions that are being asked as the researchers themselves. And this is our future CBM program. So this looks kind of like this, where we have a central node. In this example, this might be us at the University of Alberta, and then we might have freshwater solutions, and then another group, and another group, and another group running water samples. These ones might be in Alberta, this one is in Michigan, and then these might be you guys running samples or collecting samples, and this happens everywhere else too. And so all this feeds together down this way, and then it all feeds back this way as well. And that's sort of the idea of how these networks can be put together. And CBM, as I've already alluded to, has been around for a long time. And so you've all probably been part of a community-based monitoring program if you've used tools like the Secchi disc, things like a hydro lab, collected water samples in bottles, or maybe done a zooplankton tow. What we're suggesting is that this can look very different, and it can be unified around a single method, which is qPCR, and that method can allow you as community-based partners to answer questions that typically has been in only the purview of a centralized laboratory. And that, I think, really gets to this idea of democratizing water monitoring. It doesn't force you to have to contract water sampling out to a contract lab or to a state lab. You can do it yourself with this kind of a tool. So I want to talk to you about qPCR for a moment, because I think this is something that is important for everyone to get sort of on the same page. So I'm going to give you a quick biology lecture. So in a water sample, when you collect it, you have a whole bunch of organisms in that water sample. Lots of them are going to be microscopic, bacteria, things like that. But you also have multicellular organisms that you also can't see, and sometimes you'll have something you can see as well. All of my organisms for this slide are going to be swimmers with Cercaria, because that's my passion. In each of those organisms is DNA. So every organism has DNA, right? The DNA in my little picture here are these little lines, okay? My point here is that if you zoom in on those organisms, some of them have very common DNA. So some of them have DNA like this red one that's in every one of our organisms, right? That DNA is obviously not a good one to use if we wanted to find just this one here highlighted in yellow, right? Because if we were to just try to detect that red piece of DNA, we would detect every one of those different organisms, right? Organism differences being distinguished by color here. But then you'll note that some of them have unique lines. So our red one that's highlighted in yellow has this piece here that none of the other organisms have. And so the first part of designing a test that allows you to amplify a very specific target out of a water sample, which is very, very complex, is that you need to know a lot about the DNA of the organism you want to find, and a lot about the DNA of the organisms you don't want to find. Because you have to basically design a test that allows you to only find that one thing, okay? So what we do if we're going to start doing this process, and that background information, I'm not going to talk much about that right now, because that's a whole other thing about genetics and bioinformatics and stuff like that, which we don't need to get into. Just trust that as we accumulate more information, genetic information about organisms, we can become much better at doing this. And this has been why we've started to see many, many more qPCR assays evolve over the last few years, is because we've learned a lot more about environmental organisms from a genetic perspective. So what we do when we want to amplify that DNA is we take our water sample and we pop open all the animals that are inside it, all the organisms, we break them open and we free their DNA, and then it all mixes together into this big heterogeneous pool of DNA. Keep in mind that the one that we want is this little green one right here. So when we want to amplify that piece of DNA, what we do is we need to know